We will go ahead and get started in the interest of everyone's time. It is my pleasure to introduce our guest today for our Cancer Cachexia seminar series. We are so delighted to have with us Marilia Seelaender, who is a professor at the University of São Paulo in Brazil. And she's going to talk to us today about beyond the muscle tissue crosstalk and cachexia. And I'm so glad that she chose this title and to talk about this, because she is one of the people who has really pushed our field forward towards using human samples, towards really investigating how tissues connect and interact, how cancer affects other tissues, and how tissues talk to each other. So a little bit about Professor Seelaender. Before she gets started, she did her bachelor's, her master's, and her PhD at the University of São Paulo, which is where I told you that she is. So she has returned home. She also did her postdoc at the University of Oxford in the UK. She's also done some research fellowships in Germany and in Spain. And so when I was looking into her CV, I was really impressed that she is a Livre Docencia, which is a Brazilian title that is given to people who are both really outstanding teachers and outstanding researchers. And so I talked to a Brazilian postdoc in the lab next door, and he assured me that this is a really big deal. And so thank you so much for joining us today. Professor Seelaender is a board member of the Cancer Cachexia Society and has been working really hard to help us grow. So thanks so much for joining with us. I should remind you all that please put your questions in the chat. And then Professor Seelaender and I will have a bit of a discussion, and I'll read them to her at the end of her talk. So without further ado, thanks so much for joining us.
So Erin, thank you so much. This is an excellent initiative that you have, these talks, and I've been attending some of them, and it's really interesting. So it's my pleasure and my privilege to be here talking to you, and you were too kind when mentioning my background. I mean, Livre Docencia is something that many people have here, and it's a very tiresome exam that you have to undergo when you're already old, so it's very boring. Maybe it takes 3 or 4 days, but anyway, we are obliged to do it, and I did it, but it doesn't really mean that we are outstanding. It's just like a proof you can you have your own line of studies, and you teach as well. So thank you very much for being here with me. And as Erin mentioned, I want to talk a little bit about different aspects of cachexia. And we all know that muscle has been the main target of cachexia in studies. And we know that today we do not only look at muscle mass, but also at muscle function, so fatigue and decreased strength, which are present in cachetic patients. But there are much more than that. There are many other clusters in which cachexia presents an effect. For instance, in caloric intake, which is regulated by the central nervous system and by hormones from systemic organs. So there are changes in systemic, in the central nervous system, for instance, with hypothalamic dysregulation leading to anorexia. There are GI symptoms. We have catabolism, what we call metabolic chaos, which ultimately will lead to loss of organ function. This is a very poorly explored still aspect of cachexia. When I think of the liver, for instance, the main organ that determines the metabolic fate of many substrates, it's still poorly explored in the scenario of cachexia. We do have, as I said, central nervous system and also peripheral nervous system changes, which are very important. And inflammation, right, which is something that everyone knows today that underlies most of the symptoms of cachexia. So when we think of this paraneoplastic syndrome and we look only at the muscle, I hope you can see my little error here, my pointer. You see that you're missing a lot, because as in this nice illustration by Søren and that works with a group of of Mauricio Behiel Diaz, we can see that cachexia is much more. It's affecting many different organs and compartments of the body. For instance, the brain, as we mentioned, the liver, the one that I said that is still fully explored. And the immune system, of course, responds to the presence of cachexia, the GI tract, even the microbiome, which certainly shall be addressed in the future. The bone is affected, pancreas, and so on. And the adipose tissue is today envisaged as an organ which can actively contribute to cachexia. So there are some organs which will have an effect that may lead to death of the patient. So the white adipose tissue wasting, the muscle wasting, heart wasting, and circulatory changes may really compromise survival in patients. So today, I will not talk about all these aspects because that would be too much, and also I don't have the expertise for that. So we are going to focus especially on the tumor itself and on the white adipose tissue, and in the conversation between these two different tissues. So when I mention conversation, then, you know, there's a lot of exchange between these two compartments, but they are not alone in the organism. So whatever we are discussing today here implies that there is not only direct conversation between the tumor and the white adipose tissue, but this is also there is also other organs that are interfering with this crosstalk and are actually regulating this crosstalk. So it's not really just the tumor and the adipose tissue. It's the adipose tissue in the context of cachexia, bearing in mind that all these other compartments will be taken apart in these aspects that we are going to be addressing today. So when we think of adipose and tumor, we see many possible relationships. So one thing that has been known for a long time is that inflammation of the adipose tissue, such as you have, for instance, in obesity, can lead to an increased risk of tumor promotion. So inflammatory output of the adipose tissue can affect cells in different organs and promote, such as this one here in pancreas as an example, promote the origin of tumors and the progression of tumors. When the tumor is already there, then this conversation gets even more complicated because the tumor itself will produce factors that will affect the adipose tissue, which in turn will also secrete factors that will come back to the tumor. And both compartments, both the tumor and the adipose tissue, will have an important impact on the systemic and central compartments. So as Erin was mentioning, and she was kind when she said that, we have had the incredible luck of having access to human samples. So we have worked with animal models of cachexia for maybe 30 years, we still do. But lately, maybe in the last 15 years or so, we had the privilege to start some work with some surgeons from 4 different hospitals. And these nice colleagues, they really contributed in a way that we can not really be grateful enough, because we have access to samples of patients who have different types of cancer. They may be cachetic or waist stable, but most of the time we have the blood, but we also have tissue samples. So this is really something that is not so common, and we are very grateful about that. So to work with these patients, we had first to face a problem, which would be we don't actually have a definitive diagnosis criterion for cachexia. So what we did is, and this is the work of my student here, Rodolfo, he designed a software with an algorithm so that we could try to find who was cachetic and who was weight stable among our patients, and also try to grade cachexia into pre-cachexia, cachexia, and refractory cachexia. And these criteria that we employ are largely based on the work of Evans in 2008, but of course modified. So the first thing that we thought is this major question, right? So while you have people who have the same tumor diagnosis and their tumor is classified within the very same stage, why does one of these individuals develop cachexia while the other one remains weight stable? So that's a main question that was intriguing us. So back in 2015, Joanna and some more of her colleagues started looking at this problem, and we compared then the tumors from patients who were cachetic and who were weight stable. First thing that she found is that really cachexia presents no correlation with tumor stage. So indeed, you could have patients who were cachetic with initial stages of the tumor, initial disease, and some of the patients with advanced disease would not present cachexia. So we were intrigued by that, and we went further. So in this publication, what my students did was to have a look at the tumor secretion pattern and the tumor infiltration pattern. So we know today that it's been long known actually that the tumor is not only comprised by tumor cells, right? You have also immune cells, you have endothelial cells, you have fibroblasts, and different types of immune cells infiltrating this tumor. And when you compare the tumors of cachetic cancer patients, and these are GI tract cancer patients, and the tumors of those who are weight stable with the same tumors, with matched tumors, one thing that we found is that there is a decrease in the number of these M2 phenotype macrophages. So it's this relationship, this ratio between M1 and M2 macrophages can be associated with the inflammatory status of the tumor. So in this situation here, the cachetic patient has a more inflammatory environment than in comparison with the weight stable patients. So they further look at the pattern of secretion of these tumors from weight stable and cachetic cancer patients. And what we reported is that indeed there is increased TNF secretion, for instance, by tumors from cachetic patients as compared to weight stable counterparts, and of chemoattractants of many different types. So I just bring you here an example. And so we proceeded with this kind of study. And Gabriela, who's a postdoc in the lab, she produced some evidence showing that, for instance, in the patients who are cachectic, there is not an increased amount of myostatin in the circulation, for instance, or there is, even if there is a little bit of an increase in the circulation, this amount of myostatin that can be detected is not coming from the muscle itself, but rather from the tumor. So the cachetic patient tumor is capable of secreting larger amounts of myostatin, for instance, than the weight stable patient tumor. So this is already something that implies that the tumor is actually doing something to distant organs, and in this case, the muscle, for instance. And another factor that is important in cachexia is irisin. So we know that from our studies with animals, that there is an increased amount of irisin in the plasma of cachetic animals. And these authors, they also mentioned that there is an increased expression of irisin in the adipose tissue of the animals which are cachetic. And this study here points out that in cardiac cachexia, you also have an increased amount of irisin in the circulation of the patients. So again, my postdoc, she looked at this aspect in our patients, and what she found is that this irisin is not coming from, it's not really a myokine in the cachetic patient, but it's coming from the tumor itself. So why are these tumors so different? What can explain these different results, this different pattern of secretion? This is the work, recent work has just been published this week of another student of mine, Renata Castro, and we have, well, her project is having a look at what is autophagy like in the tumors of these two different types of patients. So this is a review that she published now, but we are pursuing actual experimental data on this aspect as we speak. So autophagy is a process that is very important, of course, for the function of every cell and tissue. And within the tumor microenvironment, it's important also because when you have autophagy, you will have, or functioning autophagy, will have delivery of antigens, and then you have increased anti-tumoral response. Right, so you will have, for instance, decrease in glucose metabolism by the tumor. You will have increased oxidative stress and more anti-inflammatory pattern in the microenvironment of the tumor. And we do not have excessive inflammatory response that could contribute to cachexia in terms of factors being secreted by the tumor in the circulation. However, when you have compromised autophagy, and that's what we are looking at, and it seems indeed that this is the case, this will not occur. So we will have increased and enhanced inflammatory response in the tumor microenvironment that will propagate to the system. So why would that occur? We hypothesize that when we have balanced autophagy, we will have higher purity tumors, and then we have really normal levels of cytokines, but I mean controlled levels of cytokines coming to the systemic circulation. But when you have unbalanced autophagy, so when these proteins that are part of the system are compromised in cachexia, you have different cell types here, increased inflammation, and these will reflect in the secretion by the tumor of a higher amount of pro-inflammatory cytokines leading to cachexia. So we are still pursuing this data, and I hope we can let you know about that by the conference. I hope that we can meet and face-to-face to talk about this data. So this is one aspect, but we wanted to have a look at, we wanted to have a more comprehensive view about what is going on in these tumors, why they are so different. So Joanna, who's now a postdoc in the lab, she is carrying out some proteomic analysis of the tumors from cachectic and weight stable patients of our cohorts. And as you can see here, or maybe not because this is too small, she found some differences, which we are now looking at with a lot of care. So the main differences here, which I would like to emphasize, would be in hypoxia and heat shock proteins and chaperones as a whole, and also in oxidative stress and inflammation. So the tumors from cachectic and weight stable patients, they are different in regard to these aspects. And these are in published data, so I'm showing some of them to you just as an appetizer. But what Joanna has been finding is that, for instance, oxidative stress is increased in the microenvironment of the cachectic patient tumor, and there is an increase in heat shock protein 70 expression, for instance, in presence. And very interestingly, she has seen that, or she's found that these differences that we've seen between tumors from cachectic and weight stable people, they can be related with weight loss, for instance. So here in this panel, we see here what happens to in regards to the correlation between, for instance, the presence of IF, so hypoxia-induced factor, and different other factors, and weight, and also weight in cachexia. So when we look at this, we can see that there is a marked correlation, for instance, with hypoxia and oxidative stress, which can be seen here in cachexia and cannot be spotted in the weight stable patients or tumor. So apparently there's a different regulation going on in the microenvironment of these tumors, and hypoxia is playing a very important part in this scenario. And so if chaperones and HIF are different in these different tumors, what is the meaning of that? And is this propagated to the circulation? Is this carried out? Is this communicated to the rest of the body? So this is a work of Zhang and colleagues in 2017, and they worked with different cell lineages, tumoral cell lineages, and they found something very interesting, that you have increased levels of in the medium that these cells are incubated in, increased number of extracellular vesicles. So these chaperones, HIF and whatever, may be being carried not as a soluble form in the plasma, but being carried within these vesicles, these extracellular vesicles that are shedded by all kinds of cells, and importantly by the tumor cells as well. So for instance, here, as I mentioned, they studied different tumor cells, so with the pancreas lineage being the highest in secreting these extracellular vesicles. And they also mentioned that there's a pattern, a temporal pattern, in regard to the amount of extracellular vesicles. And they found that there is an increase in HSP70 in the circulation of the animals which are cachectic. But this increase is associated with the elevated amount of extracellular vesicles. So when you inhibit them, you inhibit by an interference RNA use, so you cannot really produce the extracellular vesicles. You have this you have a decrease in the amount of HSP70 and the circulation, and also of HSP90. So the proposal of this work, the hypothesis of this work, is that the tumor cell is producing these extracellular vesicles, and this is very important today. We know about to prepare the niches for metastasis. So extracellular vesicles have been implicated in a series of processes in regard to tumor biology, and these vesicles will come to the circulation and will reach the target organs. Here, as an example, we have the muscle. So what kind of extracellular vesicles are we interested in? There are many different types, and they are classified according to their size. So we have microvesicles, and we have exosomes, which are our interest now. And as I said, these vesicles, they're implicated in a series of processes, and exosomes in particular, which are between 30 and 100 micrometers, they have a different cargo from the other vesicles. So one of these described cargos is chaperones, right? You have this HSP70, for instance, and 90 that have been described in the work of Zhang in a great amount within these extracellular vesicles, which are of this size and therefore called exosomes. So they can carry a variety of different factors. And they are now being studied. And for instance, we have this recent work of Jay and colleagues who showed that one of the microRNAs that is very important in inducing browning, for instance, in animal models, in cachexia animal models, this microRNA is carried by exosomes. So you can see here exosome production by this model, by the tumor of this model, and it's the content of these exosomes that are released is enriched in this microRNA. This is a specific microRNA that is associated with UCP1 expression, for instance, in the adipose tissue. So we got intrigued with these results, and we wanted to have a look at what was happening in our patients as well. So are these things that Joanna and my other students found being carried by exosomes, or are they just soluble and in the circulation? And a former postdoc of the lab, Marinho, he first measured or found what was the number of vesicles that were in the circulation of cancer cachetic patients or waist-stable cancer patients. And you can see here that there's a great difference already. So there's an augmented number of exosomes in the circulation of our cachetic cancer patients with, in this case, GI tract cancer. And Joanna, now working this year, she found that the exosomes of cachetic patients are enriched with HSP70. So in our patients, we have that the chaperones, which can induce modification of muscle metabolism, as suggested by Zhang and colleagues, are being carried within exosomes in the cachetic patient. And these really relates, and this exosomes and the amount of exosomes and the amount of these chaperones filled exosomes relates with C-reactive protein content and with body mass index as well. So there is an association between the amount of factors that are being carried by these microvesicles of this size, exosomes, and what we see in cachexia and cachexia effects. Furthermore, she's also seen or also reported that these exosomes from cachetic cancer patients, they are also enriched with inflammatory cytokines. So it's not only microRNAs, not only chaperones, but also inflammatory cytokines, which are being carried by this microvesicles. So for now, we've spoken about what the tumor is doing in regard to the periphery, right? We think that this tumor is secreting different factors because they're, as proved before, there is a different pattern of secretion between the tumors of cachetic and the waist-stable patients. These secretion can be carried, or the secreted factors can be carried in exosomes or not, and they will reach the adipose tissue, where they will promote probably lipolysis. So to prove this point, there's been this study of Zager and colleagues in 2016. And these scientists, what they showed is that indeed there is increased in pancreatic cancer patients of glycerol release from the adipose tissue. As you know, this is the measure of lipolysis in the adipose tissue. And so there's a big difference in the release of fatty acids and glycerol in pancreatic cancer patients as control patients. And the number of exosomes is also augmented in these cancer patients as compared with the controls, right? So when you look at that and you inhibit exosome secretion, you can see that there's a diminishment of the amount of glycerol that is released. So there is something that is coming or is present in circulation that will cause lipolysis, and this factor is associated to exosomes. So we've found that indeed there is a decreased, of course, lipid mass and delipidation in the adipose tissue in rodent models and also in patients, with decreased perilipin content in the adipose tissue of cachectic cancer patients, for instance. And it may be that this is also occurring due to this communication that is promoted by exosome-derived factors. In the adipose tissue, is thus very affected in cachexia, and I spoke about that in the conference of the Society for Sarcopenia and Cachexia in December, I guess, right? So we've demonstrated that when we compare the adipose tissue of cachectic patients, of waist-stable cancer patients, and of control patients, we see fibrosis and infiltration of immune cells. So fibrosis is going on here, and there is an increase in the amount of TGF-beta and also of TGF-beta pathway related proteins. So this is the work of Michele, and she's found that the adipocyte per se can increase the amount of secreted TGF-beta 1 and 3 in this case in the cancer patients. So there is a major rearrangement of the tissue in cachexia of the patient, but this can also imply that there will be reflexes in systemic terms. Another thing that was found, and that was previously, was that, as I told you, there's a major infiltration. There is major infiltration of inflammatory cells in the adipose tissue of cachectic patients when you compare with waist-stable patients, and there is increased, and I will not talk about that at the moment, but there is increased secretion of various inflammatory factors. So the adipocyte per se can increase the secretion of inflammatory factors that will contribute by gaining the circulation to inducing inflammation in different organs in the cachectic patient. And one thing that we noticed when this work was being carried out is that hypoxia again is important in the process. So the inflammasome that is activated within the adipocyte is being so in response to hypoxia stimuli. So when we think of the adipose tissue, then, okay, so we know that the tumor is a bad thing, then it's secreting factors that will really elicit horrible effects in the whole of the body and by gaining the circulation and affecting distant organs. But what about the adipose tissue? So we say that it's secreting TGF-beta, we say that it's secreting inflammatory cytokines. What about the adipose tissue? Normally we would say, okay, waste it, there's no problem, it's a fluffy, cute little thing. But as the bunny of the Monty Python Holy Grail, it's not as kind as one would think. So there is the fluffy thing, but it's very, very aggressive somehow, and it's causing problems that will affect the entire organism, even the central nervous system. So the adipose tissue here is not only a victim of cachexia, but it's also a very important part of it. It's important in its contribution to progression of cachexia and sustenance of an inflammatory status. So we know, and we've known for a long time known that because of obesity-related studies, that adipose tissue secretes a variety of factors which may greatly affect health. And so we have looked at some of those. I will not go through these, but I think one important thing that has to be taken in account is that fatty acids per se can be a problem in this scenario, because some of those fatty acids that are being shedded by the adipose tissue in response to inflammation, local inflammation, caused by signaling coming from the tumor and other organs. Some of these fatty acids that are being released will reach distant organs and cause inflammation and problems in these organs too. So for instance, in this work and Han and colleagues in 2018, you can see here there is an association between serum free fatty acids concentration and IL-6 production. So it's a very linear correlation here, and also in early cachexia with TNF alpha. These changes in the progression of cachexia, but still IL-6 is an important association with the amount of free fatty acids that are in circulation. But all kinds of fatty acids are the same? No, we don't. We know that this is not the case. So there's a difference of the types and in the proportion of the different fatty acids in the circulation. So Murphy and colleagues, they reported in patients with advanced cancer that there was a different amount as compared with the controls of polyunsaturated fatty acids, and especially of EPA and DHA, which were decreased in these advanced cancer patients. So this sort of implies that there is a modification in the proportion of these fatty acids. And we know that saturated fatty acids are more inflammatory, of course, than the polyunsaturated fatty acids. But so far, nobody had had a look at cachexia per se. It was advanced cancer, but not really cachexia. So we recently published a paper with Katrin Radloff and Daniela Riccardi, who were former PhD students, in which they showed that indeed there is a difference in the circulation and also in the adipose tissue. I'm not going to show this now here, but of the proportion of polyunsaturated, unsaturated fatty acids, saturated fatty acids. So you can see here is a normal person, a control, who in our study are people who come for gallbladder removal, for instance, or hernia, and the way stable cancer patient and the cachetic cancer patient. So you can see here that polyunsaturated fatty acids, for instance, are very decreased in the circulation of the cachetic cancer patients. So towards the end now, we can see that, all right, the tumor is doing things here, is secreting a lot of factors that are affecting all the organs. The adipose tissue is also secreting things, inflammatory factors. Triglycerides are being broken, so fatty acids are coming here and are also interfering with inflammatory status in all the different organs. But not only fatty acids are important. So recently, we took part in a collaboration with the group of Marilia of the Helmholtz Zentrum in Munich. And in this study, they have worked with mice and different models of cachexia in mice. And they have found that there are many differences in regard to other lipid species. So, for instance, ceramides are very changed in cachexia. You can see here that in the animal models, you see a lot of differences between the weight stable or the normal animal and the cachetic animal. And we contributed to this study by having a look with them or offering them the samples to address what was going on in the cachetic patients. And here you can see, for instance, that some types of ceramides are also changed. And ceramides are very important in controlling many different processes in different organs, right? So not only fatty acids are doing something bad here, are doing something strange and being secreted by adipose tissue, but also other lipids take part into this. Well, but okay, so the adipose tissue is affected by the tumor, but does the adipose tissue affect the tumor? And to have a look at that, we have to address the peritumoral adipose tissue. So this is a very different adipose tissue from the other ones, from subcutaneous or visceral adipose tissues. And there's a lot here going on, and you can see that the communication is really complex. I'm not going to go into all these details. You want to kill me at this point already. But you want to be the rabbit of the Monty Python film. But you can see here, for instance, but things that are being secreted by the adipocytes that are close to the tumor or infiltrating the tumor or surrounding the tumor may have effects on the tumor, on tumor biology and also on tumor secretion. So when we look at this specific tissue, peritumoral adipose tissue, we can find that there is a decreased, augment of inflammatory inputs. So these, when we compare in the weight stable cancer patient and the cachectic patient, we see that inflammatory input of these adipocytes to the tumor is increased. And more recently, Marilia has also been carried some studies with stem cells in these different adipose tissue depots, because there's a great heterogeneity among adipose depots. And one thing that she says is that while in the subcutaneous adipose tissue, there is a decreased amount of stem cells in the cachectic patients as compared to both weight stable and the control patients. With the peritumoral adipose tissue, it's not the same. So actually, you could even have a little bit of an increase. This is an old slide, and now we're finding more about this. And finally, I want to just tackle a little bit about browning, because this is the missing part of it, and and there's been a lot of talk about it. So this has been a consistent finding in animals, right? Rodent models of cachexia always show that there is some, at least to some extent, browning of the white adipose tissue, and that could interfere with cachexia. But of course, I must remind you, and today I think it's Darwin's day, so evolution and things like that, that we have a different biology from rodents. And while for us, brown adipose tissue is not so prominent, for rodents it plays really a major part in regulation of metabolism and hormones. So despite the fact that this has been a consistent finding in animal models of cachexia, we have failed so far to find the same thing in patients. And an interesting work has just appeared that, so it's Leah and colleagues from 2020, showing that what we could have here is not really a UCP1 dependent browning of adipocytes. So the final adipocyte, maybe in humans, would not be like this, but something like that. So the actual capacity of heat promotion and therefore energy consumption would not be caused by the increase of UCP1, uncoupling protein 1, augmentation in the tissue, but rather by different pathways such as is suggested here. Anyway, we would have to be so lucky to find a patch from the amount of tissue that the surgeon gives to us, in which this browning is occurring, so that we could really prove it's occurring. So I cannot say that there is no browning, but so far in the little samples that we are granted, we have not seen any real any uniform difference towards the defect or the sign that there is browning. And this is not different from what other colleagues have mentioned. So this is a recent publication by the group of Richard Skipworth, and they have looked at the subcutaneous versus visceral adipose parts, having found that the visceral seems to be more responsive to cachexia and even produce some factors that could be used in the future as markers. So this interleukin, for instance. But importantly, they found that, yes, there was a sign of inflammation in the transcriptome, increase of inflammation in the cachectic patients as compared to cancer weight stable patients, decrease in adipogenesis and increase in inflammation, but no differences in regard to the transcriptome that would suggest browning. So to end it and try to summarize it, I just found this today, this publication. It is a really challenge to pronounce the name of this nice Polish colleague, but Kaszprask, maybe. So I found this today as I was waiting my son to have a minor surgery. And it's very interesting here, the figure that this paper presents is very interesting, because it shows the dimension of the interaction, of the conversation of the different compartments in cachexia, right? So you have the muscle here being affected, but you also have the liver, you have the different compartments, the tumor is secreting actively different factors that will cause inflammation of tissue, tissue rearrangement in distant sites. But you can see here that there's a special mention to the adipocytes that are close to the tumor, and they really have an important part here, secreting factors that will influence the tumor and also the systemic circular organs, or the distant organs. And if I could add something to this figure, is that it should also include the backtalk from white adipose tissue to the tumor and to the different organs and as a whole. So thank you very much. I speak a lot, I know, and I try to be synthetic, but I think I'll always fail. And these are the people who collaborate with us. Of course, we do not do this alone. And these are our sponsors. And that's my contact address, should you be interested in knowing a little bit more about this relationship we spoke about. Thank you so much.
Well, thank you so much, Professor Seelaender, for a really wonderful talk. And because you mentioned it, I'll say Professor Seelander, to give us the talk today, has persisted through both rolling blackouts and a emergency trip to the hospital with her son this morning. So we're really grateful for your persistence in your talk today. For those of you with questions, go ahead and start putting them in the chat, and I will ask them. But I get to start. I have a question about if you feel like there might be differences in people who have excess adipose tissue and might have more inflamed adipose tissue versus individuals that have, healthier adipose tissue. Sometimes we talk about people who are healthier fat and healthier, heavier people. And I'm wondering if you have thoughts about that.
Yeah, that's very interesting, because there's been a lot of debate about obese people having cachexia or not. So I think it's very clear today that we really have obese people, even morbid obese people, who can be terribly cachectic when having cancer. But I think that this has to be addressed in two points, maybe. One thing is, like, we have to speak about different depots of adipose tissue. So if there is already ongoing inflammation, where is this of adipose tissue? Where is this going on? So if it's a subcutaneous adipose tissue, probably this is not going to be something that will worsen cachexia or the response to the tumor and the induction of cachexia. But maybe the visceral adipose tissue has a more important role in contributing to this cachexia-associated inflammation. So if you have visceral obesity, it's my suggestion that this could be worse, just very much like the COVID response as well. We are studying adipose tissue input or contribution to the cytokine storm in COVID patients. And what we find is that if you have already ongoing inflammation, systemic chronic inflammation, maybe because you're obese, or maybe for any other reason that could be related, the response is exacerbated. So yes, I think when you have already inflammation in the adipose tissue, that's bad. And the other thing is, when you may be obese, but if you're active, maybe this is not so important. I mean, physically active. So when you're exercising, even if you have more fat than you should in terms of, regular body composition, if you're exercising, the input of exercise, the control, the regulation imposed by exercise on inflammation and on the function and metabolism of different organs would block the effects of increased inflammation, local increased inflammation in the adipose tissue, for instance.
Yeah, I'm curious if you think that if some of the sexual dimorphisms that seem to be we seem to be recognizing more also might have to do with where adipose tissue is stored. As I'm sure men and women store adipose tissue very differently.
Yeah, so you see, with the women, it's more subcutaneous adipose depots, right? So, and even the response to cancer, you see, for instance, breast cancer is the one that has the least, the lowest number of cachectic patients, and Indeed, I think it's really where this fat is deposited that will have an importance there. You can see that even in animals, it's so different. Different adipose deposits will grow differently as in terms of hyperplasia or hypertrophy. It's really not the same organ when you think of it.
Yeah, I definitely agree. A couple of questions from the chat. Professor Castelli says, thanks for a very nice talk. You propose that hypoxia in the tumor microenvironment is involved in a paired autophagy and resulting high-grade inflammation. Did you check to see if HIF as well is vehicled to other tissues? So have you been able to find HIF in other tissues through the extracellular vesicles?
Oh, yeah, we're doing that now. So Joanna, the one who was interested especially in HIF, and she's going to Oxford. I mean, she's on the way to Oxford. She's just stopped in Germany now because she's working with Sir Peter Radcliffe to have a deeper look into hypoxia-induced processes. So it is indeed our hypothesis. There is increased HIF expression in the tumor. These will elicit inflammation and oxidative stress, which can be communicated to different organs. But we have not so far measured the amount of HIF that is being transported in exosomes. So that we are looking at the moment.
Another question, is there any data about activity or inactivity versus patients who develop cachexia versus patients who maintain their weight? So is there any association? We know exercise is good for these patients, but do we have any association data about that?
Right. We have this big study that we seem never to be able to publish because there is always something else that we're doing. But when even this autophagy study that we are carrying out is related with exercise as well, because we are comparing the tumors from exercised people to those of sedentary patients. We have shown in different conferences, we have published in animals the effect of exercise that really downregulates inflammation and especially inflammation in the adipose tissue. So you don't have this tissue rearrangement, you don't have infiltration anymore when the animal is exercising, and you have restoration of metabolism. So you decrease inflammation and recover metabolism when the animals are exercising. We did this with patients in the last 10 years. So we developed a protocol for endurance exercise. The patients walk on a treadmill 5 times a week. Some of them will even run around 7 kilometers an hour on the treadmill. And the results are very similar to those that we obtained with animals. So we have really down regulation of inflammation. We have recovery of metabolism. We have increased in body weight. We have increased muscle mass, and and even decrease in tumor burden. These are treatment naive patients. The only thing they are doing is exercise, and the tumor decreases size as well. So it's of major importance, all the effects of exercise, and there are so many. And when you can argue that in animals, okay, you're just looking at a physiological, pathophysiological situation. So the factor increases because, I don't know, myokines are going on. There's a nervous output. There's hormones that are being changed by exercise. But in humans, you also have the psychological part. The person has the diagnosis of cancer and, oh my God, I'm going to die. I'm losing weight, but there's no return. And then suddenly, the person starts to feel better and can climb, let's say, sets of stairs that this person was not able to do before getting the diagnosis. So this impact, psychological impact, is also very important. But anyway, physiologically, only the effects on inflammation and hormonal regulation and metabolism are there. It's really good that we exercise, and it can be endurance. It doesn't have to be strength exercise, because we are not only aiming at the muscle or mitigating muscle loss, but we are aiming at decreasing systemic inflammation, and endurance exercise does that.
Yeah, I think there's certainly a lot that we don't know about the psychosocial aspects of both exercise in these patients and in cachexia in these patients. A question about how you think about the continuum of cachexia, and if you think that we might be seeing some of the same changes in these patients who are just on the cusp of being considered cachectic, by the definition we have, or might, still be considered pre-cachexia, versus patients who have maybe late stage cachexia and lost a lot of weight. Is there a way your lab thinks about that?
The thing is, Vicky Baracus once said in the lab to me, she said, well, maybe given the chance, every patient, cancer patient would become cachetic. But I'm not so sure about that because, well, at first, there's this pre-cachetic population is very hard to define. That's the main point. So we did, we tried something with some physicists, and they developed a machine learning protocol. And in which we fed all the data from the patients. And so we had our human classification of cachetic, non-cachetic, maybe pre-cachetic. And the computer was what we call, what they call, the physicists say, it's a non-learn. Well, the computer did not learn what was cachetic and non-cachetic before. So the computer just separates the patients according to what it thinks is more reliable. And by doing so, we found that in the population of patients that we considered weight stable, there were some of them who were falling in between. So maybe they were pre-cachetic. And then we went to try to find why the computer chose to put them in a in-between zone, and it was already inflammation, and it was a different pattern of inflammation. The thing is, the cachetic patient, when you look at the C-reactive protein, for instance, in the circulation, you can find that it's high, not enormously high, but high and constant. While in the non-cachetic, the weight stable, the true weight stable, it's within normal or a little bit high, but not really something that calls attention. But these in-between population, they had a peak of CRP and the circulation. So it was much higher than the cachetic, let alone from the controls. So it suggests that there is something like a trigger that makes inflammation go up very quickly. Then it comes down and stays high, but a little bit lower. But there is like a shock. And when we exercise the patients, we see something similar, but on going towards the other end. If you exercise the patients for 3 weeks, you have not much change. But then, in week 3, something happened, and you can see that all the factors that we measure go up very pronouncedly, very exacerbatedly. And after that, it goes a little bit down, but you can see, for instance, IL-10, it goes up like that, and then it goes back, and it's higher all the time. So exercise is also eliciting something that is like a trigger. It's like the patient starts to respond to exercise stimulus as a anti-inflammatory thing as well. So it's hard to work with the continuum, but there is a continuum, I'm sure about that. And there is something that triggers the worst response at a certain point.
Yeah, I think that really speaks to, perhaps the individual nature of cachexia, right? I think we're learning that different tumors and different patients just simply have different cachexias.
Yeah.
And different root causes, perhaps moreover, of their cachexia. And lastly, a question about macrophages. You found that you fewer M2 macrophages in your cachectic patients. Is that surprising, both from a fibrosis and TGF-beta perspective, but also just from those tend to be the tumor-promoting macrophages?
Yeah, there was a figure in my former slide which was really about that, because we would say that M2 macrophages, if they are decreased, that would benefit the tumor by somehow. So it's a paradox that I cannot really now explain why it happens, but it is there, and it's consistently there, because we have not only worked with this GI tract tumors, but also with lung tumor and pancreas tumor, and we can see that the same is going there. Maybe it's not really the amount, because this is just a parameter, the actual number of M2 macrophages, but the relationship and proportion with different immune cells that are also present there in infiltration. So these lower purity tumors seem to be more cachexia eliciting than the other ones.
Yeah,
and pancreatic tumor is very, of very poor purity.
Yeah, lots of stroma and lots of immune cells in that microenvironment, right? So, well, I think we'll stop there in the interest of time, but thank you so much, Professor Seelaender, for a really wonderful talk and stimulating discussion. I would like to remind everybody that our March seminar will be on March 10th. It will be at 4 p.m. Eastern Standard Time, so convert that to whatever time zone you're in. We're going to go a little bit later and try and get that part of the world involved. Our speaker will be Professor Chris Kost from The Ohio State University, and he is going to tell us about elevated immune checkpoint inhibitor clearance in cancer cachexia patients. So we're going to talk a lot about therapy in cachectic patients and how their response to therapy is different than patients who are maintaining their weight. So thank you so much again, Professor Seelander, and thank you all for joining us, and have a great rest of your day wherever you are.
Thank you very much again for the invitation. And I'm here if the people that want more questions can always write to me in my mail. Thank you very much. Thank you for your interest. And
thanks, everyone.